So I think what I'm going to do today, well, and I'll introduce myself a little bit more. I'm at TGen, the Translational Genomics Research Institute. I'm in the Division of Integrated Cancer Genomics. I actually studied at the University of Toronto and moved to TGen to basically learn about genomics and got into epigenomics. And so during my postdoc work, I spent quite a bit of time learning about DNA methylation and other epigenetic changes. And what I want to do today is talk to you about DNA methylation and DNA methylation data, and then ways to access the data that's available on the TCGA, and then just briefly talk a little bit about ways to analyze it, because we'll get into the required tools towards the end of the talk, and also then go through the workshop and get some practice on how to do some of these things. So before I start, I'm just curious to know how many people here are interested in epigenetics. Of course, not surprising in the least. How many people here have worked with methylation data in the past? Okay, and how many people are interested in using the TCGA in the methylation data available on the TCGA? All right, so hopefully this will be a useful sort of first step for you if you haven't already accessed the data. So the outline of my talk is to give you an overview of epigenetics. I think that in general, DNA methylation and epigenetics is still not very well understood and lags behind the understanding of some of the other fields that we know so well, such as transcription and sequencing. So I'm going to give you an overview of epigenetics and DNA methylation. I'm going to discuss the methylation assays and discuss some of the reasons why the platforms were selected by the TCGA. And those would be the Illumina Bead arrays, which are probably the most popular high throughput array-based methylation assays available. We'll go to the TCGA website and we'll talk about how to query the data and download data and how to interpret that data. And we'll discuss a little bit about analysis, and I'll show you just a little bit about some of the data that I've done. So also to sort of give you a little bit of so because I'm at TGen, I've generated my own methylation data. So there's you can get the data for free, or you can spend a lot of money and generate it yourself. Having said that, that just simply is to say that I've done these assays myself, and so I understand the platform, and so hopefully can give you a little bit of insight about the platforms. So epigenetics, in some ways, can be described in a number of ways, but these are the most common definitions of epigenetics. The modern definition of epigenetics is that it's modifications of the DNA or associated proteins other than DNA sequence variation, which is the key point here, that still carry information content during cell division. So it's a heritable change. It's not a genetic sequence structure change, but it's still heritable. And it's supposed it's also has a lot of impact on gene function. Epigenetic processes are genetic modifications that affect gene regulation by changing chromatin conformation. So 3 different ways you can kind of look at it. I also always say they're chemical modifications to DNA, which is in many ways true. So the epigenetic code consists of 3 main components: DNA methylation, histone modifications, and non-coding RNA, for those who didn't know that. And DNA methylation is covalent addition of methyl groups to 5' positions of cytosines in a CpG dinucleotide. The reaction is catalyzed by DNA methyltransferases. In mammals, 70 to 80% of all CpGs are methylated. Most occur in repetitive elements or regions of low CpG density. We also have heard very, very commonly of CpG islands. These are CpG-rich regions. They're generally found in promoters, but not always, and they're generally unmethylated in normal tissue. CpG islands methylation of CpG island is associated with delayed replication, condensed chromatin, and inhibition of transcription, but not always. That's just kind of like a very basic explanation of what methylation can do. It's critical for normal development, and we all know that aberrant methylation is associated with cancer progression. So the assays. So I want to take you through, there's so many different types of assays available for DNA methylation. They can be bisulfite based. Methylation specific restriction enzymes can be used as a way to detect methylated sequences, or you can actually IP methylated DNA. If you can use either of those basic assays or basic principles towards array-based or deep sequencing methylation methods. All right, and there are a number of them. As the sequencing and the reagents are becoming more readily available and are getting better, methylation sequencing is going to become very popular as well. But till now, it has lagged behind all the other sequencing methods because the library preps and the data analysis has it's very difficult, very complex, but it's definitely catching up. So when you're picking the assay, you have to consider a number of things. How much sample do you have? What is the quality of the sample? Obviously for TCGA, sample quality won't be an issue, but sample throughput, gene throughput, bias in the assay. Are you going to be preferentially isolating or detecting islands or not? The workflow and the cost. Okay, so there are a number of factors that have to go into selecting the assay. And actually, before even anything to do with the TCGA, from my own personal experience, I went through this process and landed on using the Illumina-based arrays because it met all of those criteria in the previous slide. The Illumina-based arrays are bisulfite-based, and they're an excellent way to analyze DNA methylation. So for those of you who do not know, and this is really, really important if you really want to grasp DNA methylation data, okay, you need to understand what bisulfite conversion is, and this is the TCGA data for methylation is based on bisulfite conversion, so it's critical, I think, to understand what it is. So this is a very, very clever way of converting an epigenetic code to a genetic code, because remember, the sequence is the same. CPG, whether it's methylated or not, has the same sequence. So how do we convert that to information that can be utilized by genetic assays? And the way to do that is to bisulfite convert the DNA. So methylated DNAs are protected from bisulfite conversion. Bisulfite conversion converts every other cytosine in the genome and unmethylated cytosines, whether they are associated to a guanine or not, to a uracil, which subsequently gets converted to a thymine. All right, and so you now have a SNP discrimination. You have a C to T discrimination, and you can design probes preferentially to methylated or unmethylated DNA, and that's how you can, and that's basically the basis of the Illumina assay. So the Illumina bead arrays are not new. They've been used for genotyping long before methylation came into the picture. In fact, they only started using them for methylation, I think, in 2007. So they're very, very new assays, relatively speaking. And it's a high-throughput single nucleotide polymorphism genotyping system that was adapted for DNA methylation to basically be able to identify bisulfite-converted genomic DNA. I think it's brilliant, actually. So, and the assay is really, really robust. The platforms have evolved. The Golden Gate was one of the first assays that I used because I started doing methylation, DNA methylation studies in 2008. So it was still a fairly new platform at the time. And at that time, you were only able, still a lot of loci, able to detect about 1,500 CPG loci across 800 or so cancer-related genes. That's based on Golden Gate chemistry, which is very different from what the TCGA now is using, which is based on Infiniium chemistry, which are the human methylation 27 bead chip, which recognizes 27,500 loci across about 15,000 genes. And now, I guess it's been about a year and a half or so, they released the 450K Infiniium methylation bead chip, which is basically genome-wide, 450K CPG loci across most genes. And the reason why you will see both types of data on the TCGA is because this wasn't available when they started. All right. So when you're considering the TCGA data that is available for the human methylation 27 bead chip, this is what you're capturing. You're capturing 27,000 loci across 14,000 consensus coding sequences in the database. You're capturing miRNA promoters, cancer hotspots, multiple targets per gene. Okay, so unlike gene expression array, which is you could have, a couple of probes per gene, this is slightly different because the location of the CPG in the gene can actually have can be very different. So when you're looking for concordance between CPG, especially if they're very, very far apart from each other where the probes are designed, you're not necessarily going to look for concordance between the probes because the location of the probe has a lot to do with what you might expect to see in terms of data. You get single site CPG resolution. This allows high sample throughput, and you require as little as 500 nanograms of DNA. Okay. How does this differ from the 450K aside from the fact that it has many, many more probes, exceeding 485,000 methylation sites? It covers about 99% of RefSeq genes. Only 90% of those, by the way, are common to human, the 27,000 chip. It covers about 96% of CPG islands with an average of 17 CPG sites per gene. So you'll see when you query your genes, you might get multiple probes, and you may have to go through every individual one to actually be able to get an idea of the methylation status of a gene in a given sample set. And the sites are distributed across promoters, 5' UTRs, first exon gene bodies, and 3' UTRs. The annotation is much more extensive. It's not available on the TCGA website, but you can download it from Illumina. So it covers islands and shores and shelves, and it's basically just distances from transcription start site. CPG sites outside of CPG islands, so many, many non-CPG island associated loci are included. Differentially methylated sites that are known, so it will annotate the arrays annotated for those areas that have been previously described as differentially methylated regions and other tumors. CPG islands outside of coding regions, so a lot of repetitive elements are included. miRNA promoters again, and disease-associated regions identified through GWAS. All right, so it's very, very extensive in its annotation, and so it's a lot of data to deal with, and you kind of just have to pick and choose what is going to best suit your needs, or maybe you can start with a simple question and work your way to include some of these other things. And I'll tell you about some of the ways and things that I've done in the past in terms of dealing with the annotation and what you're looking for potentially. So the process starts off with bisulfite conversion. All right, you whole genome amplify and you fragment your DNA. You hybridize it to a chip that pretty much looks like this in real life. And then that hybridization is followed by by single base extension, okay, which provides your floors that give you your measurements. The process takes about 4 days because what's not included on here is the bisulfite conversion. So if you consider that day 0, it takes about 4 days from start to finish if you ever want to try to do this yourself. And I should also say in terms of cost, it works out to about $330 or so per sample. So it's not unreasonable. So for the human 27K used Infiniium 1 chemistry. Now, this may seem a little bit over what you need, but it's actually important when you're considering the analysis, especially for those bioinformaticians. At the end of the day, I'm still going to give you a simplistic sort of explanation of how to analyze the data, but if you really want to dig deep and do kind of fancier types of analyses, it's important to consider the different chemistries on the array, and you can get what probe has what chemistry from the annotation file. All right, so the human methylation 27K and some probes of the human 450K use Infiniium 1 chemistry. So what does that mean? It means that there are 2 different probes. One recognizes unmethylated DNA, one recognizes methylated DNA. and the probe binds to the full CPG. So the single base extension happens after the binding of the full CPG. So the single base extension happens to the adjacent CPG locus. All right, and the different probes, whether it's unmethylated or methylated, for a given probe has the same floor. So you're not comparing green to red for Infiniium 1. You're comparing red or green. But if there's a mismatch, the probes won't bind, and that's how you really end up being able to discriminate between a methylated and an unmethylated, because the methylation probes, the probes that recognize methylated DNA won't bind to unmethylated DNA sequences, if that makes sense. The Infiniium 2 chemistry, which actually allows for the coverage, the depth that is made available to us by the 450K chip, is because of the Infiniium 2 chemistry. And the difference here is that the single base extension occurs, next to the C. All right, so, and these are two colored, but it occurs next to the C. So it's a slightly different binding, and the single base extension happens in a slightly different location. So there aren't probes that recognize methylated and unmethylated. There just won't be binding to the methylated sequences if it's unmethylated, and vice versa. So the important thing to recognize about Infiniium 1 and Infiniium 2 chemistries, and Illumina sort of made this known at the very beginning, but a lot of people very, very interested in really learning about different ways to analyze the 450K methylation data meaning more the bioinformaticians than the biologists, have put a lot of attention and emphasis lately into how the two probes behave. And they behave differently. They actually have different distributions. So the dotted lines are Infiniium 2 chemistries, and obviously have a distribution across methylated to hemimethylated to fully methylated loci. But if you look at the solid lines, represent Infiniium 1 chemistry, and Infiniium 2 chemistry is by these dotted lines. So this could become a problem for certain specific things. If you're kind of comparing locus to locus, if you're comparing one locus between two samples, it's not so much of an issue. It might become more of an issue when you're kind of going to compare locus to locus and you're looking for more, extensive types of analyses. You might have to consider and worry about, these different distributions. But if you're just looking at a differentially methylated locus between two samples, maybe not so much. So after you hybridize and you stain, the bead arrays are scanned using Illumina iScan technology, and the raw data is extracted using R-based methylamine. I guess I'm saying that a little bit wrong. Package 25 to calculate the beta value, DNA methylation score for each probe. Okay, but you can also use, this is what the TCGA uses, but you can use Genome Studio software, which is what I use to extract the data as well. Okay, now this is obviously the next point here is going to be very relevant to you if you're looking at the TCGA data, because the data is presented in or in some of the levels of data availabilities is presented as a beta value. Okay, and that's basically the level of DNA methylation at each locus. Okay, it's scored as a beta value, and it's calculated as the methylated signal over the sum of signals. Okay, and you get a number ranging from 0 to 1, 0 being the most unmethylated, 1 being the most methylated. Does it mean that that 0.5 is hemimethylated? Not necessarily. You have to consider your sample and the heterogeneity within the sample, and what are the different samples are going to be contributing to that score. Okay, so obviously if it's 0 or 1, you really have a clear signal, but rarely do you get complete 0, and rarely do you ever get 1, but you might get close to it. Everything in the middle could mean a lot of different things. So you have to just keep that in mind. And when you're sort of describing your data, don't describe it as hemimethylated, it's 0.5. That could mean a number of different things. So I think you've all seen this by now. This is to access the data, you have to go to the TCGA portal. This is the website. If you don't already have it, I'm sure you do. And essentially you just go here, launch the data portal. Okay. And it's really not that difficult to download the data, which is really great, unless you want to download high-level data. Now, what we're going to do towards the end is spend a little bit of time navigating this website. There is a lot of information, and pretty much everything you need to know about the data is available. You just have to kind of dig for it. It's not necessarily very easy to find, but it's there. They have kind of like the NCI Wiki, so you can get a lot of information. You can get frequently asked questions. You can really, figure out what some of the things mean. There are some tutorials on there. There are guidelines. It's really, really a great website. There's a lot of information on it, but you just have to really navigate it and spend some time digging to get answers that you may not know. One of the things, if you go, once you launch the portal, you're going to have some tabs up at the top. There's some tools. This tools tab is going to take you to all kinds of really good links for data analysis and other things. And about the data, you can get a lot of information. And one of the things that it does is you can go to platform design, because there are so many different types of platforms. It's kind of evolving, right? As platforms become available, etc., they get added or adapted and things like that. But you can click on any of these and it takes you to the company's website, right to the source, and you can get information about the cost, and whatever else. You can get all kinds of tech notes. Illumina, if you really want to dig deeper into these platforms, has some really great information about these arrays. And if you create an Illumina account, you'll be able to download all the things. One of the things you may want to download, especially if you're more in the bioinformatics and you're going to do some broad level analyses, you might want to download that annotation file for the 450K and the 27K chips. Okay, so one of the things you can do from the homepage of the data portal site is you can decide to do two things. You can download data or you can query data. So it really depends on what you want to do and what your question is. If you're interested in just a subset of genes and you don't want to, and you don't have the tools or maybe even the know-how necessarily to work with the downloaded data, and also the time, because it's going to be a little bit more time consuming. The data, when you download it, is not ready to go. You kind of have to work with it a little bit. But if you want some quick answers, if you want to look at some other, you're not just interested in methylation, but you're interested in copy number as well and gene expression, you can kind of get information about a subset of genes really, really quickly. So the query the data is, a really good place to start if you're interested in just, your favorite gene. And then on this site also, it shows you the status of the cancers types that are available, so you'll be able to see what you can query and what you can't. Not every single tumor type has the same data sets available, so it might be a little bit of a hit and miss, but I think it's pretty good. And then you can also do the download data. So if you're going to query the data, you can just click on query the data, really easy, and that's going to take you to this page. You can select your disease type over here. All right. And then you can decide what you want to do. All genes, I wouldn't recommend it if you want to querying the data for all genes. You should probably just download the data if you're going to do that. But if you, are interested in a chromosomal region and you're interested in maybe the methylation status of a certain area of the chromosome, that might be a good place to start. It'll be easier to mine than having to mine it yourself, because essentially it's mining it for you when you query the data. Or you can just insert a gene list, as few as one gene, as many as 100. Okay, so there's a lot of things, and you can export the data after that and still work with it. Participant list, I've never done anything other than all participants, but depending on your needs, you may have a subset of samples that you're interested in. And then, here you can actually do two things at once. You don't have to just select DNA methylation. You can select DNA methylation and copy number and gene expression. So there's a lot of different combinations you can do. Again, it requires a little bit of manipulating. It's not just completely effortless. Once you click these buttons, you still have to put some, a little bit of, use a little bit of analytical tools to really extrapolate the information that you need. I've tried to work with the correlations a little bit. I didn't have a lot of success with this one, so I probably won't go over it with you. But for DNA methylation purposes, there's a couple things that I would like to point out and emphasize. So there's a pull-down menu, and you can just click on human methylation 27. For some reason, you can't query 450K data. So if you're interested in 450, at least maybe for certain tumor types, I don't know what the extent of all the tumors and but for GBMs and I think even breast cancer, you can only query human methylation 27 data, and it'll take you to a page that looks like that. So here are the hits. So these are the different probes. 100% of the samples met your criteria. You can click on each one of these probes and it'll give you the methylation value for each sample. Okay, so you're not just selecting. Unless you're interested in highly methylated samples, then you can change your criteria to 0.5 or above. So you can, kind of quantile your data if you do it like that, or you can get just all the data at one point. You can export all the samples or all the information. Here's information on just a reminder of what you did. If you go back and you do the same thing, but click on the tabs above, you'll get the samples. This may not be too applicable for necessarily what you're doing here, but if you look at pathways and MGMT doing some of the similar, it'll just show you what are the statistically significant associated pathways with that gene, and it just gives you an idea of what it's involved in potentially. Now, I think I was saying that you could look at gene expression. So if MGMT methylation is the gene expressed or not in a sample that has high methylation or low methylation. So when you do that, and you can query them together, this is just a snapshot from another query the data that I did both methylation and gene expression on, and you can get log2 sort of full change information or z-scores. It just depends on what's available. It's not, the same things aren't available for every tumor type. And the other thing that's not similar when you're trying to sort of do these types of correlations is that not every sample is the same. So you're going to have to match the samples yourself. And as an example, here you see that I think it was sample 9 has a methylation value of 0.66, and methylation value 10 has even a higher methylation value of 0.786. So those are fairly strongly methylated at that locus for MGMT for those patients. And if you look at samples 9 and 10, the gene is not expressed or has fairly low expression. So there is an inverse relationship right there, and you can do some of those things. Don't always expect to see a strong methylation associating with loss of expression. That is not going to happen, and if you're going to look for it all the time, you're going to be disappointed, all right? There are all kinds of things that can happen when you're considering methylation and gene expression data, and so far in my experience, we've done a lot of these analyses, the correlation seems to be in the 25% range. So just keep that in mind. Okay. So downloading the data is another way you can access data, whether it's methylation, gene expression, sequencing, whatever. And the steps are going to be pretty much the same. There are two things. There are going to be two levels here, data matrix and bulk download. Personally, I like the data matrix. It's a little bit easier. It's a little bit more interactive. You can filter the filtered sort of input. Bulk data download is just going to give you so much information, a lot of, and it might be a little more cumbersome to work with, but it'll get you to the same endpoint. I'm just going to show you the data matrix feature here. Okay. And you can read a little bit more extensively about what are the real differences between the two. At the end of the day, you'll get to the same endpoint. It's just easier with the data matrix, I think. There's also maybe not so necessary for the DNA methylation because, but you can, if you want a large amounts of data or access to really raw data sets in large numbers, you may have to get permission to access the portal. For a lot of things that most people will need, you probably can just do the publicly available databases. So very simple. You just click on data matrix. When you, I guess I should have maybe put this on another slide, but there are 3 different levels of data. Okay. They'll matter differently for different data sets. I'll tell you about what matters for methylation. Unless you're a bioinformatician and you really want to look at signal intensities and normalization issues, I wouldn't even bother with level 1 data when you're looking at methylation data. It's going to give you, for the human 27 chip, it's going to give you the raw signal intensities or the IDAT files for the 450K. That's just a lot of information. You don't need to do that. I never do that. I always go with the output of Genome Studio, which is the beta value. If you have the beta value for most things that you want to do, you're golden, you're fine. You know, you can extract other things from the beta value if you're interested in M over U instead of M over U plus M, those types of things for the bioinformaticians in the audience. But this is level 1 data is just too raw for the methylation component. Level 2 data and level 3 data are pretty much the same, with the exception that level 3 data is a little bit annotated. It tells you where the probe is and the gene name. You probably want to use level 3 data because it gives you some annotation without having to go back and dig it up yourself. So I would definitely recommend level 3 data. And I personally, if I were to do this, I would ignore level 1 and level 2 data, but that's because that suits my needs. It may not suit all of your needs. But these are the differences. And there's also metadata information. We'll talk about that in a second. So if you go to the data matrix again, you can select your disease type. You can download clinical data as well, but we'll just talk about DNA methylation data here. If you know what batch, or if you want to work with small numbers at a time just to get an idea, don't download everything. You won't be able to actually download everything anyway because it only allows you a certain amount of downloading capacity. But you can work in batches. If you have sample IDs, you can put them in. If you're interested in both 27K and 450K data, you can highlight both of those, or you can highlight one, but it doesn't matter. Even if you highlight both, I'll show you why, because it's an interactive tool. So you can end up filtering what you just filtered, okay? The data level, again, you can put all 3. It doesn't matter because you can still filter out, but, level 3 is fine. Of course, if you want data available now, you click on that. Then there's different types of tumor samples. So you can get tumor-matched pairs, or you can get tumor and normal unmatched. You can just select what you want. Now you may have to go higher because you may not have enough samples if you are really narrow in your filter. So you may always want to start with just clicking everything and then selecting what ends up becoming available so you at least get something in your search result, if that makes sense. And then again, you click and you're going to get to a screen that looks like that. And so here, what did I select? The 450K and the 27. So here it both showed up, but you see how there's an indent here. It's because you can select either everything, or you can end up just selecting, the 450K data, and then you can even just, do control, kind of select, and you can select whatever you want from what's shown. And the legend is here for what the sample type is. So this is a tumor that has a matched normal. The matched normal sample is going to be in yellow, and you can just follow the other legends. It's pretty self-explanatory. You can always go back and edit your filter. If you want to go back, it's much easier to always use the tools here, because if you end up pressing back on these TCGA, it takes you back to home pages and things like that. So just follow the tabs that are available here. So once you do that, you're gonna, get your samples are gonna be available to you like this. You can click or unclick anything that is there. If you don't want the metadata, because you don't really care which lab and whose name and all that stuff, you can click it off, but it's fine. You can end up selecting only the matched pairs if you want. So again, it's very interactive. You can filter the filtered. But the point here is that you're only allowed 70 gigabytes of download at one time. If you need more than that, you have to do it in batches. And then all you do is you enter your email address, and a link to the data gets emailed to you. It's gonna look like that. You just click the link, the hyperlink here, and it's really fast. It happens like in seconds. It's pretty good. The metadata file, as I was saying, all it is just high-level data showing you, what are the, who's the lab and what is the experimental factor, just a lot of information about what your downloaded data, who generated and all that stuff. The file manifest just gives you details by sample. It's just kind of a summary of what each sample is. There's really not a lot here. So so you're gonna download the file. It might take a little bit of time to download. And depending on how big your file is, it's a zip file. It'll unzip it for you. And you can just, go down the folders until you get to your files. So when I mean by processing the data, I mean processing because it's not all the samples are not put in one Excel sheet for you. Ready to go, here you go. Delta betas are there. No, they're individual files for each sample. So you then have to sort of work with that information. All right, and if you're downloading tumor samples and normal samples, I think you heard about this yesterday, you have to be cognizant of what the sample is. So the sample ID for the matched samples are going to be the same, but the number preceding that is going to tell you if it's tumor or not. And there's a really good, I think you can even get this from frequently asked questions, what the barcode is, what does that long sample name mean? And I think you went over this yesterday. But for this point here, what you want to know is that tumor types range from 0 to 9. So anything from 01 to 09 is a tumor. And anything that has a 10 or 9 to 19 in that sample name preceding the sample ID itself is a normal. So you'll be able to discriminate between tumor and normal that way. So this is what the level 3 methylation data looks like. It's going to be all the probes. This is, and we'll work with 27K data because the 450K data is too large to work with, and it's just easier to work with 27K data today. But this is the same sample ID down across the entire 27,000 probes. Okay, and this is the probe name that Illumina gives. This is the beta value associated with that probe. This is the gene name, the chromosome number, and the position, the chromosomal position. So this is what level 3 data is. If you only did level 2 data, you're only going to get beta value and maybe probe name. Well, and probe name. So that's why it's just nice to know. It's nice to have this extra stuff here. You can do some high-level analyses like this way. Or maybe high level, not meaning complex, but simple types of analyses, just using Excel actually. Okay. But I did want to also point out very important things about the meaning of NA and methylation data. It differs based on whether you're in level 2 data or level 3 data. And the reason why you have NA there could be a number of reasons. If it's level 2 data, human Infiniium methylation 27 level 2 data, it's basically p-values that detection p-values that were greater than 0.05. So basically p-values that didn't pass the noise to background ratio. Did I say that right? It wasn't above the background, okay, enough to be considered a good probe to use. All right, so level 3 data has a number of different reasons why you would have NA. Most of these are common to both 27K and 450K, but 450K has an additional thing. So it contains a known SNP in the region. So if I was doing the analysis, I may not exclude these probes. They're excluding them for you here, so you don't have a choice. Okay, it contains repetitive sequences are not included in the data. All right, even though if you had the data yourself, you would keep that data, but you can't with the TCGA data. It's filtered out. They're not uniquely aligned to the human genome, build 36. They span known regions of small insertions and deletions, and they have a SNP within 10 base pairs of, I think maybe even 50 base pairs of the interrogated locus. Okay, the annotation file tells you, if you generated your own data, the annotation file, which gets updated as new SNPs are entered into the database, allows you to include them or not include them, and there isn't a consensus in the field as to whether you should include them or not, because there's no real evidence that within 10 base pairs or even 50 base pairs, that you even have the SNP in your sample, or that it's going to affect binding of the probe. It's just predicted to affect binding of the probe. So it's kind of up to you to decide. When you're working with a cancer methylome, you've got so many thousands of hits that removing those is not going to be, just to keep filtering and increase the prioritization of your final list. Filtering those is probably going to just make your life easier in some ways. You should never ignore data that you filter, because you might still have to go back and learn, because if things just miss the filter point, there still might be some additional probes that belong to a gene that is looking interesting, things like that. Okay, methylation analysis, there's a number of things you can do. You can, the easiest sort of thing to do is perform a differential methylated analysis to identify differentially methylated loci. And the way I define this, there's slight variations to this. It's kind of like deciding whether you want to use a twofold change for expression or you want to use a 1.5. So it's a little bit arbitrary. 20% or a delta beta of 0.2 seems to be about standard, what people are doing. But it's a locus that differs by more than 20% or a delta beta of 0.2 when compared to normal samples or whatever your reference group is, and survive statistical significance. So you have to do non-parametric is usually the standard type of differential methylation statistic that you do. And essentially that gives you a delta beta value. If you're working with Genome Studio, is anybody here working with methylation data in Genome Studio? No, okay. So if you're working with Genome Studio, the new version of the program calculates delta beta for you if you tell them what your samples are. You can also look at the distribution of loci. You can learn a lot of really cool things, like some of the TCGA publications don't necessarily calculate delta beta values, but they give you heat maps showing like how certain loci vary between molecular subtype of breast cancer, for example. So you can cluster your data. You can look at regional differences. You want to maybe look at promoter methylation of a gene and how does that change compared to the gene body. You may also want the data to say, well, this is differentially methylated. Is it expressed or not? Could this be a potential tumor suppressor? There's a methylation hit. There's a mutation. Maybe I've identified a novel tumor suppressor gene. So it's an important level of data that can really be, that can help you prioritize your findings, that can help you learn more about the biology. It's sometimes a little bit of not sure what to do with methylation data all the time, because there isn't necessarily an immediate functional output or outcome, but it can help guide you towards your final list of genes that you want to work with, all right, because it's just another level of change. So what you would do with all those files, you would open them all up, Again, if you're working with a bioinformatician, you can have them do this and write programs and softwares. I can't do that, so I just do it sort of this old-fashioned way. Just open up the files, create multiple tabs. I create big giant Excel sheets where I put all the samples into one Excel sheet. And, you know, so instead of having now, and the good thing is that the order of the arrays is the same for every sample. So you can easily just copy and paste you know, the columns out of one text file into a master text file. So here you have the normal sample and the tumor sample. Did I get that right? Yeah. The tumor sample and the normal sample from the 11, okay? And I calculate a delta beta. So it's basically this minus that equals delta beta. It's really easy, just a simple Excel formula. You can basically carry that all the way down. There are your NA probes, so you're just gonna filter those out. And what you're looking for here for the delta beta is anything greater than 0.2 or less than minus 0.2. Again, this is arbitrary. This is what I do when I analyze the data. And those are going to give you what I call differentially methylated loci. Keeping in mind this has not passed statistical processing yet. I didn't do that here because you can't do non-parametric tests on Excel. So I just to show you how to just do it simply, you will have to first pass these two columns, have them go through statistics. Obviously, if it's one sample to one sample, you can't do that. If you're looking at a matched pair, you can't do that very easily. But if you're going to group the samples, you'll just create an average of all the tumors, an average of all the normals, have that pass statistical significance, some kind of multiple correction testing, which is really difficult with the 450K. And so I just want to give you some ideas of some of my own data that I generated, you know, based on, this is actually using Golden Gate data, so it's kind of old, but it gives you an idea of why the annotation is important and why it's important to know whether it's CPG island or non-CPG island. These are just, you know, basically counts and frequencies of what is occurring by, this is multiple myeloma showing sort of progressive hypomethylation. The way you, depending on how you calculate your data value, a negative number implies that the tumor has less methylation than the normal. If it has an increased or a positive number, it has more methylation than the normal. Okay, and again, that's just by convention, and it's the easiest way to kind of explain it, to always do it that way. And so, you know, the difference between CPG island and non-CPG island, you can see that the hypomethylation is occurring mostly on non-CPG islands. So this is again, this is much more extensive annotation now. You can do this with shores and shelves and north shores and south shelves with the 450K data, but this is sort of just a basic overview. If you cluster, this is what you can, you know, you can get. So there's, you know, basal-like breast cancers that show, you know, lower methylation compared, in fact, this is basal-like breast cancer to the brain that show, you know, overall much lower methylation compared to some of the other subtypes, which was actually consistent with the TCGA methylation data on breast cancer that just recently got published rather. You can show here, you can plot the methylation data, just take all your values and just do a box plot, and you can see the distribution. You can get statistical significances, values between the means. And these are just some examples of how to, you know, some of the ways you can analyze the data that you generate. This is another sort of heat map of 450K data showing, you know, tumor compared to normal, adjacent brain. And so that's it. So I think we're right about on target for an hour. The practical is going to be, if you haven't done that and if you're interested, just to spend, you know, 10 or so minutes navigating the TCGA website, getting to the portal, spending a little bit of time querying data, just pick some favorite genes, and then also spending some time downloading data. I have, if you want to play with the Excel files, I can save what I have and just give it to you to work with, or you can actually go through the exercise of downloading it and working with it. And Kim Bussey, who's also at TGen, who's going to be giving the next talk, is going to help with the practical, and we'll come around and we'll help you guys, and you can ask as many questions as you want. And her talk, which she's going to be focusing on gene expression, is going to be a nice complement to this, and she's going to show you some really cool tools on how to, you know, use some of the web-based analyses that are out there to analyze specifically gene expression data web-based analyses for methylation data are really not that good, and they're not that prevalent. So you have to kind of rely on Excel. I use GeneSpring, or our good bioinformatician friends who can write all kinds of fun programs and get some cool outputs. So I think with that, I'm going to end and just ask if anybody has any questions before we maybe take a couple minutes break and then just move into the exercises. So it's a little bit of free roaming, because I think you can just very easily get to these sites. I'll probably go back and just maybe leave my slide to the website, so you can copy the let's go back even here. You can copy down the web page here and just go to it and kind of navigate from there. A lot of you might already know this, and forgive me if this is already too basic or something, but so if you go to here, I can go, I want greater than or equal to 0.2, or, because if you say and, you're going to get 0 hits, and less than or equal to minus 0.2, and it filters all of your probes out that don't meet that criteria. So here's potentially a list of differentially methylated loci, and we went from 27 or so thousand to 4,600. Okay, we're going to do some additional filters. We're going to remove the X chromosome and the Y chromosome, and we're now down to 4396. There are so many additional filters that you could, apply, like statistics and multiple correction testing, which can help bring your list down. And then you can do things like, you can sort by, by gene name, which it's already sorted here. And you can just always go back and just modify that filter if you're looking for things that are really high. You don't want the threshold to be that sort of, to lack that stringency. You can even look for the things that are really high. Then you can count the number. Okay, so here we're actually going to sort by value, because we're going to have some. So here's something that's completely almost unmethylated compared. So this probe is unmethylated in normal, and it's completely methylated almost, in the tumor. It's hypomethylated, basically, a very, very hypomethylated. And you can count to see how many hypomethylated probes compared to methylated probes is going to give you a trend, or is this tumor type tending towards hypermethylation or not. Where, for you to do some deeper dives into the data, you need the annotation file, so you can figure out if it's a CPG island or not, and just where it's located. So, but you can see here that these changes are not associated with gene symbols, so you may want to actually filter that out and just go to scroll to the bottom here and just remove, I guess it's doing it in alphabetical order. Get rid of the NAs. So our gene list is down to 491. And you can, some things might MUC1, I think I saw something else. The TEMM genes, this is breast cancer data, so some things might already start to make sense. Claudin 8 is known to be important. FFAR2, this dreadful Excel problem that we have. But so here, just FGF1, so FGF1 is hypomethylated in tumor compared to normal. So this is really easy for you to work with the data. You don't need to have programming skills, and you can just do some really good, easy things just this way.
So, Bodour, do you want to mention why you removed the sex chromosomes out, just in case anybody, it's not obvious?
Imprinting. If you don't know, which you won't, the gender of the patient, there are gender differences in methylation patterns in the sex chromosomes between male and female. So obviously you're going to have a mixed pool of samples. If you know that this patient's a female breast cancer, you may not want to remove those because you might actually be able to get some differences related to loss of exon activation and changes in imprinting. But otherwise, if you're grouping samples and there's going to be males and females in the group, which can happen for breast cancer 1% of the time, you might just want to filter those out. So how do you get your file into Excel? That's a really good question. It may not be
and how do you get it into Excel without
obvious.
Screwing up the gene names?
Please ignore my my messy desktop. Okay, messing up the gene. Okay, we'll do that as well. So you're gonna right click. These are text delimited files, tab delimited files. Sorry. You're gonna right click. You're gonna open with. This is a little bit different sometimes. Well, for PC it's the same, but my PC doesn't allow me to do that. I have to go to Excel and open the file from Excel. So if you're on a PC, you may not be able to do this very easily. But if you're on a Mac, you can just open with Microsoft Excel. Another really important point when you're working with text files and you're copying one text file into a master Excel file, it's going to allow you to do that. It won't allow you to save it as an Excel file, as a tab delimited file. So make sure you're not just clicking save without changing the file type, because you will lose everything you did. Tab delimited files are they're just one sheets. All right, single sheets. And if you do any formatting, any filtering, it's gone. So make sure that you save as Excel. Once you start building your Excel file, just rename your file immediately so you don't forget, as a master Excel file type, like an Excel file type. And so here's your tab sheet. It's now in an Excel file, so you can easily drag and drop if you have working with two screens. Like I'll just show you some. These are really Excel tricks that might help you with. You can like drag and drop the file. You don't even have to do anything. You can go to right click, move or copy, and you can select the workbook, create a new workbook. So those are some just tricks for you when you're dealing with these. So you can tell I've worked because they're just easy tricks for you, and they help sort of make the process a little bit more efficient. And then you have to go and make sure that you're saving as an Excel workbook. And usually it's going to be over here. It's going to be at a tab delimited text. Does that answer your question?
So I am gonna correct one thing.
Yes.
And
so
that is
I didn't do it.
that if you want to avoid what Excel does to gene names.
Let's do this. Although it doesn't always work, Kim.
To do that, you know, don't do that.
Oh, don't do that, right.
Do not do that. Because you need to pull Excel's wizard for importing text. Because you are going to manually have to tell Excel, be dumb about the data type. So
we're
this
gonna go to,
and it's a known issue. It happened when we went from Excel 97 to 2003, and I've screamed at Microsoft, and everybody else has screamed at Microsoft, and it's made it into the databases, and they haven't fixed it. So you're going to want to do a tab delimited text. You're going to hit next.
So you're going to basically not right click and open with. You're going to go to Excel, and you're going to you're going to find the file on your wherever it is in the directory. And you're going to go next.
You're going to go next, and it's tab delimited. You're going to go next. You're going to scroll over to where you have gene names in that column, and you're going to highlight that column where the gene names are, and you're going to say it is text. Okay? And you're going to finish it.
And if we search for December 1st, it probably won't be here.
Yep, it'll be DEC1. Where Excel is going to do that is any gene name that looks like a date, and any clone ID where they did not include the library name. The difference is going to be that when it looks like a date, it's going to convert it to a date. If you open it up, you all of a sudden see date names, and you say, oh, I forgot to do text, and you go up and you say, make this column text. It will now convert that date to a number, because that's how Excel is storing that information. and it'll give you the number, and so you've lost it. There are tools that are available that will convert that back for you if that happens. The easiest way to get out of that is just not to save a thing and open the file again from within Excel. It's much harder to get back to clone IDs. So this arises from the problem that anytime you have a clone ID in a library, row E is, you know, plate number, E, well number. When you pull that into Excel, Excel goes, oh, you're dumb, you don't know scientific notation, and converts it to scientific notation. And if you don't know what plate number you were, you can't get back. And so
I get lazy because I'm never interested in those.
those
They
are
somehow get
definitely
filtered out.
things that
Yeah,
you
but-
have to worry about.
But that's a good point there, definitely.
Can we go over that again one more time, please?
Sure, with the opening the file?
File, yeah,
Oh, sure. You mean,
into Excel.
okay, so let's just open another one. So you're gonna go File, Open. You're gonna go to your directory. So you're just gonna keep going to all the different, so all these folders. You're gonna click a file. You're gonna go Open. It's gonna take you to this import wizard. You're gonna click Next for the most part because it's delimited, okay? That's usually default. You don't ever have to change anything most times. There are all kinds of cool things you can do here, but for just simplicity here, it is a tab. You wanna basically, which means you wanna separate the data into columns. You're gonna click Next. Here, it's not, so as a default, it's general, but general usually is number, okay? And so you're gonna highlight the name of the gene column, and you're gonna convert that to text so it doesn't change it to a number or date column. And then you're gonna finish, and that's gonna open. Then you have to save this if you're gonna work with it in any way, filter it, do anything, highlight, make notes that require any type of formatting. You have to go to Save As, and here where you have tab delimited, if you just save it as tab delimited, and I'll show you why you can easily do that, because you might just do all of this stuff and the file's already saved on your desktop, so you just hit save. And you just keep saying continue and it saves it, and then you've lost everything. So you have to go to File, Save As. You have to scroll to the top, which is usually the first format is the Excel format. You may not see it when you're initially opening it up. Just scroll to the very top. It's the first file type, and you save it as an Excel workbook. If you have, you can continue to play around. If you have any questions, Kim and I will be walking around helping where needed.